all this work in this area is directed towards trying to improve outcomes for infants that are born with these sort of problems at birth. You'll find all of the specific projects detailed in the book that Megan will talk about at the end. Thanks, Megan.
Sorry, my mute button disappeared. Okay, the next speaker is Dr. Shayanti Mukherjee, and she's going to tell us about the sort of research that she does and many of the other people do within the Women's Health Research Group. Thanks, Shayanti.
Hi, everyone. I'm Shayanti. I'll be talking to you today about some of the work that we do in women's health theme. A lot of our work is laboratory based, and what you see here is some of the data, the kind of data that we produce to generate novel information about diseases such as endometriosis, adenomyosis, or fibroids. A big part of the theme also looks at endometrial stem cells, which are highly regenerative cells in the uterine lining of the womb. So we work on cell culture methods to prepare them for clinical applications, or even understanding their gene profile to see what their role might be in diseases. We also work on exosomes, which you see what role they play in disease progression, or how they can be of therapeutic value for women's health applications and diseases. And there's also a big focus on developing patient-derived models such as organoids to see in 3D, model the disease and improve the diagnostic of diseases such as endometriosis, which can take 12 years in the current time. So going on to the next slide, we also have quite a bit of focus on maternal health, so pelvic organ prolapse and maternal birth injury. And big part of this part of the research focuses on tissue engineering. So again, we're combining materials with stem cells to develop new generation of constructs for treatment for these diseases. We also look at technologies such as 3D printing and 3D bioprinting of these cells, looking at very detailed cell material interactions, and use, like Stuart said, we use small and large animal models to study how the disease progresses or how we can develop treatments. Another aspect here is to look at nanoscopic tissue details using atomic force microscopy or biospectroscopy to understand how the tissues progress into the disease and whether there's a way we can stop them. So there's various different angles and different aspects of women's health, different women's health disorders that you could actually contribute to.
Thanks, Shayanti. Okay, so our next speaker in today's session is Dr. Kirsten Palmer, and she's the head of maternal fetal medicine at Monash Health and a senior lecturer at Monash University. Thanks, Kirsten.
Oh, thanks, Megan. And apologies, here was I thinking I'll just put something very simple together that doesn't have any animation and is on one slide, and looks like it still had some animation in it. So if you just want to push the next one, so nothing needs to move around and change. All right, keep going. Perfect. Apologies for that. So my name is Kirsten. I'm a clinician researcher based at Monash Health with affiliations as well with Monash University, and I work very closely with the team within the Ritchie Centre as well. And I think this is a real highlight of both of the sessions with the Department of O&G in terms of what a collaborative environment it actually is, because for all the projects that are reported, really it does require a very extensive collaborative team effort to undertake this research. I undertake a range of different projects, and these really encompass all the way from discovery laboratory-based research through to population and epidemiology clinical research, clinical trials research, all the way through to health implementation research. But the main areas of focus that I really wanted to kind of highlight this evening was the work that I undertake in relation to placental dysfunction. The placenta is such a key organ in achieving a healthy pregnancy. And as such, it plays a really major role in a range of pregnancy complications, including preeclampsia, fetal growth restriction, and stillbirth. So within these areas, I'm undertaking discovery research to really try and understand what is happening within the placenta that contributes to the development of these adverse outcomes. And this is work being undertaken quite closely with staff within the Ritchie Centre, such as Dr. Stacey Ellery, Dr. Sarah Marshall, and Dr. Emily Cam. And then taking the findings that we're achieving within the laboratory, translating them through to clinical trials for promising new therapeutics. And to this extent, we're doing work with translating through sulforaphane, a really promising antioxidant for the treatment of early onset preeclampsia, which is a clinical trial we're hoping to commence in the next 6 months. And we're currently leading a multi-centre, multinational clinical trial of melatonin for fetal neuroprotection in the setting of early onset fetal growth restriction. Likewise, work that has really been led out of the Ritchie Centre looking at a very common nutritional supplement, which is creatine, undertaking a range of work with Dr. Stacey Ellery, where creatine holds a lot of promise for protecting the unborn baby from antenatal and intrapartum hypoxic events, which is also something we're hoping to be able to translate through to a phase 2 clinical trial in the near future. Another large area of research over this last 18 month period has also been the COVID-19 pandemic. And I think this highlights the real value that doing research can have in terms of helping you rapidly pivot and change as need be to provide information to guide future care. So together with a range of clinician researchers from across the state, during the course of last year, we developed the Chopin Registry, which is collecting information on every Australian woman infected with COVID during pregnancy. And this has been really crucial information to help to inform clinical care, but also the structure of service provision now with the current Delta outbreak. So this has been research that's been set up very quickly and rapidly translated through. We've also developed the Command Registry, which is assisting in providing real-time feedback around the impact of changes in lockdown and care access on maternity outcomes. And then I've led the work in implementing telehealth into the provision of antenatal care here at Monash Health. And this is now giving us a big opportunity to reconsider what the future of ideal woman-centred care can look like in the 21st century, and has gone on to inform an extensive research program, not only looking at clinical implementation research and the impact on clinical outcomes, but also qualitative research looking at clinician and consumer experience, and also a health economic evaluation to understand the true cost to healthcare funders. So I hope that I've just given you a taste of the diversity of research that's achievable within our project as well, within our program.
Thanks, Kirsten. Okay, and our next speaker is Fabrizio Horta, and Fab is going to tell us give us some examples of the sorts of research that get conducted within our education program in reproductive development. Thanks, Fab.
Thanks, Megan. So just next slide, please. So yeah, we are EPRD, that is the Education Program in Reproduction and Development. It's part of the Department of Obstetrics and Gynaecology. Although we mainly focus in education, we also develop research, mainly in the areas of embryology, andrology, cryopreservation, idiopathic infertility, as well as novel technologies in IVF. This is mainly the laboratory part that is developed in IVF clinics. So all the embryology behind the scenes, all the work behind the scenes in clinics to actually make babies is pretty much what we normally, most of our education is focused, but we also, we do education on it. EPRD research based on small research projects that are built into the into the coursework degrees that we have. Dr. Sally Cat will talk a little bit more about that. That is the graduate diploma in reproductive science and the master clinical embryology. However, the EPRD staff also takes some number of research projects, such as honor projects, master of reproductive science by research course, and also PhD student in the areas of idiopathic infertility, which is infertility of unknown reason, as well as sperm DNA damage, sperm selection, oocyte and embryo cryopreservation, as well as vitrification of ovarian tissue. And also novel technologies. Even recently, we start doing some research in artificial intelligence in IVF. So the next slide, please. In order to carry out this research, we partner with different institutions as well as companies. At the moment, we are partnering with the Department of Data Science and Artificial Intelligence, as well as the group of mechanical and aerospace engineer. But we also have previous and continuous collaboration with other entities, such as Ferring Pharmaceutical, Monash IVF. Currently, we have some projects available. Next slide, please. So the best way, if you want to connect with us, is use the Supervisor Connect. You will find our projects over there. At the moment, we have one project available. So I will encourage you to have a look on it, and please feel free to contact us. Thank you very much.
Thanks very much, Fab. So our next speaker is Sally Cat, and she's going to tell you about some more information about the education programs in within the education program in reproduction and development, which is within the Department of Obstetrics and Gynaecology. Thanks, Sally.
Thanks, Megan. So Fab has explained the research side of our work, which is down this arm, but for something sort of completely different, we've been looking at you doing probably honours and research. If you end up with just a degree, you don't want to go through honours, there is a pathway that you can take where you can actually come into our programs doing a grad dip in reproductive science, and you only need, say, 60% in your bachelor's degree. This can actually lead you into all sorts of things, this grad dip. It can lead you down the path of the Master of Clinical Embryology, or you can go into research. Maybe next slide, please. So the grad dip is your sort of honours equivalence. It can be honours equivalent if you achieve a D or an HD. And pretty much it's a coursework course, 8 compulsory units, and some pretty fun subjects to study, a bit of practical skills in IVF, and all these other courses here, which I'm not going to go through one by one. It has about 36 students in it this year, and we do have a high percentage of Monash students coming into this course. And it's set not at the university, but at the Monash Medical Centre. And these are our 2 wonderful coordinators. The top one is practicing what she preaches at the moment on mat leave, and below is Nicola. And they'll both be with us next year, we hope. And so the next slide, please. So this is the course that I've been running for far too long. It's pretty much a course on steroids. It's a bit of a boot camp, 72 credit points in 1 year, but you're learning heaps of practical skills all through the year. You're in the lab day in, day out, even at night. And alongside the practical, you're learning all the theory, and we're also teaching you a few research skills along the way. We've got great equipment, really well facilitated labs, and yeah, I recommend it. So I think that might be it. We won't any more. This is just all the information. I presume these notes are going to be going out to students. So if you want to know more about it, all the information's here. Okay, that's it.
Thanks, Sally. And here's a picture of the team.
Oh, yeah.
Okay, so our next speaker is Ben Amberg. Ben did his Bachelor of Medical Science Honours degree in the department and continued on to the MD PhD. And in fact, the Department of Obstetrics and Gynaecology probably has the greatest proportion of MD PhD students in the faculty. Ben.
Thanks, Megan. I might need your help just to click along to a few slides. So if you just click, I'll just get a little bit of a timeline up. So thanks everyone for having me along to chat. Hopefully I can provide a few insights into what it's like sort of on the ground level as a student. Yep, so I've got 2 more clicks, I think, Megan. I feel like I've been invited along to a bit of a speed dating for the wise. I feel like I've sort of accidentally stumbled my way in here, but this is a bit of a timeline about where I've got to where I am. So as you can kind of see, I'm a bit of a professional student. I did the first 4 years of my medical degree and then thought, hang on, I really like science and research. I'll take a year off and I'll do my Bachelor of Medical Science with honours, like Megan said. And then I was like, you know what, like lots of my friends were going off to graduate and going back to the last year. And I was like, you know what, I actually really like this. I'm just going to keep following on to what I like and what I'm really enjoying. And so I converted that BMed Sci year into a PhD, which was sort of another 3 years on top of that, and had never really looked back. Loved my 3 years at the Ritchie Centre and have met some wonderful people and had some really amazing experiences. And then this year I'm back trying to sort of finish off my medical degree, and then hopefully at the end of this year I'll finally graduate and become more than just a Year 12 graduate. And next year get a real job and become an adult and hopefully move on with some sort of research in the future if people will have me along. So this is how I've got to here. I'll tell you a little bit about my project and then I've got 3 just sort of helpful hints for the future. So Megan, if you go to the next bit. So I just want to, this video, and hopefully you can make it out from the clip, is just some pretty incredible footage from inside the uterus during pregnancy. So this baby has a congenital abnormality of its lower back, of its spine, called spina bifida. And basically, if we did nothing for this baby over the rest of its pregnancy, it would be born with pretty much lower limb paralysis. So its legs wouldn't work. It would pretty much be incontinent for the rest of its life. Now, the reason we have this clip is because we've put a camera into the uterus and some surgical instruments sort of halfway through pregnancy. And what we then do is we can stitch, or this is a clip from the US, but they stitch the spine to sort of back together. And then hopefully over the rest of the pregnancy, we prevent some damage to the spine and that baby will be born with functioning legs and functioning bladder, which to me is just incredible. Now, we sort of took this surgery that's being done mostly in the US and in Europe, and we sort of said, hang on, these are wonderful, but like, let's make sure that it's safe for the baby. So we replicated the surgery in a sort of a preclinical model, and we were sort of looking at what happens to the baby during these surgeries, what happens to its heart rate, what happens to its blood pressure, what happens to the gases in its blood. And then we had a look at it, like what happens to the placenta, which is that kind of big UFO purple thing floating in the background. And we were looking at all sort of the things that sort of go along with that. So I had a really good insight into sort of some of the large animal model type physiology that we do at the Ritchie Centre, which was really useful to work with and really cool to see it sort of directly translate across into some of the human stuff that we were seeing being reported overseas. So that was my project. I loved it. I wish you could all do my project and follow along because I had a great time. So I have 3 tips. The first one is be micro ambitious. As you can kind of see here, if you'd asked me in 2011 when I graduated Year 12, I would not have told you that these were all the things that I was going to do. Each one of those steps is just me being like, you know what, I'm really enjoying this. I'm just going to go on to this next little thing to keep following the things that I'm really enjoying. And looking back now, there's no way that I would have chosen anything else. And I'm really proud of that sort of pathway that I've created for myself, just kind of following things that I'm really enjoying. Number 2, get good at being wrong. A little anecdote, when I sent these slides initially to Lisieux, who set the night together, I actually had a spelling mistake in that sentence, which I just thought was the most amazing example of this. Over the course of research, you're going to get really good at having a thought about something and going to your supervisor with, I think this is what's going to happen, and you're just going to be completely wrong. And I think there's a wonderful art that you develop in research about developing how to be wrong. All right, and then the last one, just really super quickly, no worries, is pick the people, not the project. I think as students, we often have an idea about what we think will be a really cool project, but we don't know at all. Make sure you pick the people that you think you're going to work really well with, because they're the people that motivate you to do your work, and they're the people that are often role models for you in your careers moving forward. So pick the people, not the project, every day of the week. Thanks for having me. I hope you learn one or two things.
Thanks very much, Ben. Okay, so, and I'm just going to very quickly go through the actual options that you have if you're interested in applying for a master's or a PhD. Now, I should clarify, this is a master's by research. Sally has already shown you the links for applying for the courses that EPRD run. I just want to talk about the more traditional master's and PhD by research. As Ben just said, the supervisor is all important. I think most of us would say the supervisor is far more important than the project. So find the general area that you're interested in, and then make a time, email the supervisors, make a time to meet with them, and via Zoom, obviously at the moment, and make sure that you're a good fit with them. This is the link if you're interested in applying for a PhD. And there's an upcoming scholarship round for local students on the 31st of October. And the Department of O&G PhD graduate coordinator is Professor Caroline Gargett. If you're interested in interested in doing honours next year, we accept students from at least 3 different honours degrees in the Department of O&G. Students from Bachelor of Biomedical Science honours. Professor Mark Hedger is the coordinator on this precinct. Bachelor of Science honours. Most of our honours students come through the Department of Anatomy and Developmental Biology, and Olga is the coordinator there. Or if you're a medical student considering BMedSci honours next year, then speak to me. You can find projects. I think Jen has just popped the link to the projects in the chat. So on the School of Clinical Sciences website, you can download the Honours Research Project booklet there. As Stuart mentioned, the Ritchie Centre is part of the Hudson Institute, but also part of one of the research arms in the Department of O&G, so you can find a list of projects there as well. You can also search via Supervisor Connect, which Fab has already mentioned. So I will stop there and open up the session for questions from the audience. Just feel free to, you can just turn yourselves, you can either raise your hand or turn yourself off mute to ask a question, or you can pop a question in the chat. Thanks very much, Jim. Do we have any questions from the chat? Sorry, from the students on the presentation? That was a whirlwind tour of some of the things that we've done in O&G. I know some of you were in the previous session and might have already asked questions there. Jasmine, what are the requirements to get into the Masters of Clinical Embryology? Sally?
Yeah, sure. So you don't need to do the grad dip, but if you do, you sort of automatically get a pass into there if you get above 70%. But really, you just need honours. Any honours will do. Not really fussy, obviously, or science-based if you're on this whatever we're on. So yeah, any science-based honours, and it doesn't really matter too much about the grade. That's it.
Okay, do we have any more questions? You can just turn yourselves off mute if you'd like to ask a question directly of any of our speakers today.
I'm just putting my email in there in case you wonder whether it's a question.
Thanks, Sally. Okay, so I think, students are often worried about approaching several different supervisors, and I just want to reassure you that actually that's perfectly normal, and we expect students to approach several different supervisors, so don't be worried about doing that. The normal process is that you would email usually, as I said, several potential honours or PhD supervisors, make a time to Zoom with them, and then get that sense. And the that they will, it's an interview in each way. So as a student, you're interviewing the supervisor to see if they're going to be an appropriate supervisor for you, and the supervisor's interviewing you to see if you'd be a good match for their research group. So it's a real two-way decision. And once you do make a decision, once you get to the point where you think, okay, well, that's definitely my preferred project, I strongly recommend that the moment you make that decision, email the supervisor and sign up to them then. We certainly last year, we ran out of Bachelor of Biomedical Science honours. Not we as a department, the whole university ran out of Commonwealth supported places for BMS honours students. So if you're a BMS student thinking about honours, then get in fast is my advice to make sure that lots of students missed out on an honours place last year from Bachelor of Biomedical Science. Do we have any more questions? Olivia, Elena, Kate, Bailey, Yasmin, Wada, Anusha? Does that also apply to Bachelor of Science? My understanding is that last year there were still places available in honours for Bachelor of Science, even though we'd run out of places for Bachelor of Biomedical Science. But of course, we don't know from year to year. We don't know now what will happen this year. It's the very first year it had ever happened that we'd ever run out of Bachelor of Biomedical Science honours places last year. So I think get in early and get in fast is my recommendation. Yeah, so I think that's a similar quota issue for both of you.
Do we apply for honours through the faculty we did our major in? Or can we also apply through the faculty we did our minor in?
That's a good question. I think you'll need to look at those departments. Which departments did you do your major and minor in?
So I did my major in phys, and then my minor is dev. So I don't know if I can go through Olga because she's kind of the main contact.
You're doing BME 3082 at the moment, aren't you? Is that right?
As an elective.
Yeah, all you have to do is go to, so usually the Department of Physiology requires one supervisor to be in the Department of Physiology, whereas in Department of Anatomy, both of your supervisors can be in the Ritchie Centre or Department of O&G, like down on our site, in other words, on our precinct. Just double check the entry criteria in developmental biology. BME 3082 is one of the units that contributes towards you being able to do a major. So you'll have to just check. Actually, I'm 99% sure for a Bachelor of Science. You're Bachelor of Science, not Bachelor of Biomedical Science, aren't you?
Bachelor of Science.
I'm 99% sure that you can do it in anywhere.
Yeah, Megan, can I jump in there? It's been delinked. You used to have to do it through, if you did your major through physiology, that way you had to do your honours through physiology, but it's actually been delinked now, and you can nominate any department to do your honours through. So you can do it through, we would suggest that you do it through anatomy and developmental biology for that simple reason that Megan mentioned about having to have a sort of a satellite supervisor who knows nothing about the project.
Okay, now I've just realised that it is 6:05 p.m. If any students wanted to jump into the Department of Nutrition and Dietetics session, you should jump out of this Zoom and jump into that Zoom now. But if you would like to keep asking questions of the Department of O&G, I think most of us can hang on for another few minutes if there's any other questions you'd like to ask. Eleanor,
I've got another meeting.
Okay, thank you, Bev.
See you later.
Eleanor?
Yeah, I have a question about the logistics or the possibility of doing having like a semester 2 next year start. I'm open to starting in semester 1, but like if it was the right project, I'd do it. But is there the possibility to start in semester 2?
So that might depend on whether you're doing Bachelor of Science or Bachelor of Biomedical Science. Science. Okay. So again, because they ran last year, they ran out of the Commonwealth supported places for the Bachelor of Biomedical Science honours students, which meant they couldn't take a mid-year cohort.
Yeah.
But the Bachelor of Science, my understanding was that they didn't run out of Bachelor of Science projects. just Commonwealth supported funded places, so that I think they did have a mid-year intake of honours students, but I think it will depend a lot on the type of project that you want to do. So if Eleanor, are you currently doing BME 308 or?
Yeah, and I have spoken to a supervisor who is open, like she's very, she was keen, in fact, to like have a mid-semester start, but it was more just a matter of if the project is taken by someone starting in semester 1, then, that would be kind of just unfortunate. So, yeah, I'm trying to toss up whether I just started a new job and I have like a 12-month thing, and I'm trying to decide whether to leave that early or the 12-month contract finishes in June. So I'm trying to decide whether to.
Okay, so I think it definitely makes, and Stuart might want to jump in here, there's definitely some projects that are much better starting in February. And I think the other advantage is that if you start in February with the other students, you're part of a really big cohort. Whereas if you start mid-year, you're one of a handful of students. So I think the big cohort is good for a sense of community and connectedness. But Stuart, go on, you jump in.
Yeah, look, I completely agree with that, Megan. It's far better actually doing it because you're going through all of the different challenges of doing honours with all of the other students. So you're facing the same issues that they are in terms of the coursework. And so you can talk about it, and it's much better doing that together. Also, as Megan mentioned, the projects, like for instance, if you're doing a fetal sheep project, that is actually a bit of a detriment to do it start mid-year, because the sheep breeding season really only goes from May till perhaps the end of November. And then we have a long down period between there and January. So there'd be a long period where you wouldn't have any access to animals. So that makes it a lot more complicated. And indeed, you would have to compress your experimental side of things right down probably into that first 3 months, because you wouldn't have enough time to, once the program starts up again, maybe at the end of April, before having to submit your thesis and write up your thesis, or analyse your data, write up your thesis, then submit it. So it puts a lot of pressure on. So I wouldn't recommend it for a fetal sheep project, but any other project, it should be fine, other than the problem with, not going through with a large cohort of students doing the same thing that you are.
Yeah.
And I think you become besties. You become bestie mates with with the other honours students. You really, particularly if you are part of the Ritchie Centre, we've got a massive student room. And you really become, in a normal year, of course, when we're not a lockdown, you become really good friends with a whole batch of other students who are going through at the same time. Now, I've just realised it's the Department of Paediatrics session that's on at the moment, not Department of Nutrition and Dietetics. So you might have been keen to go to that one. Sorry.
No, only the women's health stuff. So I'm good.
So just to give you an update, we have 30 honours students this year.
Wow, yeah. Yeah, I mean, that's been very informative, and I think it's very good advice, and it's, going to, you've changed my mind a bit, to be honest. So, thank you so much for the advice.
Olivia, I saw your screen on before. Would you like to ask a question?
I did. Also, I think it's the Department of Surgery that's on at the moment.
Oh, is it?
Yeah.
Oh.
I panicked and wanted to go to the paediatrics one, but I think it's surgery.
Oh, they must have changed the audio. Okay.
I know, I understand it's probably project dependent, but what are the time commitments generally like for an honours student? So I'm kind of in the same boat as Eleanor, in the sense that I've just got a great job that I wasn't wanting to quit for honours, but if it's possible to juggle both, is that a thing, or?
So again, very project dependent. So the honours degree is classified as a full-time degree, which is technically 48 hours per week. So it's essentially, we expect you to treat it like a full-time job, so 9 a.m. to 5 p.m. Having said that, there are some projects that might require you to be there on weekends or evenings, and there are some projects that are more, particularly some of the human data projects, where actually the time's a little bit more flexible. but very project dependent. The start of the year is less busy than from about a third of the way through the year. Things start to really ramp up in terms of how busy you are. So that would depends on whether your work is only on weekends or if it's taking up weekday time.
Yeah. Okay.
Can I jump in there again? I think Olivia, talk to the supervisor, because sometimes the supervisors can work around that. For instance, we often have data that hasn't been, we've done experiments and we have data that needs analysing. So that means there's no set time commitments where you have to be in the centre. And so as long as you get the work done, then that's all we're really interested in, and plus the coursework as well, as Megan just mentioned. So just talk to your supervisor that you really are interested in, and they might be able to work around so that you can keep your job and or even go part-time. So there's all sorts of flexibility there.
Cool. I guess that's what I was wondering, if there's flexibility, but no, thank you for that.
I think most students would have a part-time job. I'm sure that lockdown has affected lots of people's part-time jobs, but most people would go through part-time. Listen, the other little thing I should have probably popped on the slides, for those of you interested, Ben, our wonderful Ben on the screen here, last year during lockdown, he and one of the other PhD students in the department developed a HUDcast, a podcast series based in the Ritchie Centre, and they're really fantastic. So if you get all the background knowledge, info, behind the scenes stories from, if you listen to those HUDcast series, and you can find them anywhere that you find your podcast. Is that right, Ben?
Yeah, that's right. We basically just got bored during lockdown when we should have been writing our thesis, and thought we just wanted to chat to all our friends that we had in the lab. So if you want, there's maybe 20, 25 different supervisors from the Hudson, from all departments and the Ritchie Centre, and that we just got a bit of a summary about what research they do, and then a little bit about their careers and what they like to do. So I don't know, it's probably a 50-50 chance that if you've got a project in mind, have a listen to get a bit of the secret background squiz on what they're really like. It's probably a good idea.
That's really enjoyable, thoroughly enjoyable listening. Any more questions? No, it looks like, oh, Eleanor, listen to one. Great.
Yeah, it was brought up at the Ritchie Centre Zoom yesterday. So then I actually went and listened to one on my walk today. So yeah, no, it's really good.
They're really funny. Well done. Okay. Yes, and it looks like several of you have been listening to the HUDcast, so that's great. See, Ben, you're going to have to start them up again, I think.
Yeah, I know. Definitely have to get them going again.
Okay, well, if there's no more questions, I'd like to really thank all the speakers today for coming along and speaking to you, and the students for coming along and listening to us. And we look forward to meeting you next year, face to face, hopefully, in the Department of O&G and or the Ritchie Centre.